All right, in Chapter 8, we are going to be talking about genetics in cancer. And you may remember from previous discussions that a relatively small proportion of your cancer risk is believed to be hereditary, about 10 to 20%, with the other 80 to 90% coming from the environment. And so cancers generally are distinguished as being either inherited, and we would call those familial cancers, the way you'll see them referred to in this chapter, versus sporadic, so non-hereditary cancers. And of course, familial cancers are inherited because you are inheriting mutations in specific genes, right? Typically genes uh, associated with cell cycle control, right? Oncogenes, or as you're going to see in this case, in most cases, tumor suppressor genes. So an example would be if you inherit BRCA1 and or BRCA2 mutations, you're going to be at increased risk for breast and potentially ovarian cancer as well. And you see in the table here, there's quite a few different cancer types that have been associated with inherited mutations. And you can see, for example, about 20% of ovarian cancers are believed to be due to inherited mutations, about 9% of breast cancers, 8% of prostate cancers, and so on. Now, I'm going to give you some examples of these inherited familial cancers, but before we do that, I wanted to do just a quick review of some basic genetics because this is going to be important for our discussion. And so just to remind you, our human cells are diploid, meaning we have 2 copies of every chromosome, and hence basically 2 copies of every gene that we're going to be discussing as well. And I also wanted to introduce this idea of penetrance. And we are kind of conditioned, if you've studied Mendelian genetics before, we're conditioned to think of penetrance of these genes and their phenotypes as being complete, meaning if you have a dominant version of a gene, dominant A or dominant B, we would expect anybody who has at least one copy of that dominant gene is going to have the dominant phenotype, whatever that may be associated with that gene, right? 100% of the people. So we would say that is complete penetrance. That phenotype is penetrating completely in that case. Same situation with the recessive allele, right? We would expect, let's say here, we would expect everybody with the recessive little d genotype to have the recessive D phenotype. 100% of those people, complete penetrance. But there are quite a few examples in the natural world where that's not the case, where somebody has a phenotype that may be different from their particular genotype. And so there's a number of different reasons for that. In some cases it could be environmental factors, but I just want you to be aware that there are instances where not everybody with a particular genotype has the expected phenotype. And that's what we're going to see with some of these cancer syndromes, is this incomplete penetrance. And hopefully it'll become clear as we go through a little bit of mechanism here why that is the case. So the first example I wanted to give you of a familial cancer is retinoblastoma. This is a rare childhood cancer of the eye, the retina, as the name would imply, only about 200 cases a year. And until about the mid-1800s, pretty much every child that developed retinoblastoma would die from that disease. You can see the big tumor in the retina here, and it would very easily spread down the optic nerve into the brain. So what changed in the mid-1800s was the development of the ophthalmoscope, that little device that shines the light, bright light into the eye, and would allow doctors to identify these tumors earlier. You can see what it would look like to a physician here in the picture on the right. And so once it was identified, if you could catch it before it spread, had a very high cure rate now. But what was interesting is now of those children that survived retinoblastoma, their kids had a much higher chance of developing the same early childhood cancer. as much as 50% increased risk. So this was really the first example of what was believed to be a hereditary cancer, right? Oh, there seems to be some genetic component here instead of just exposure to, some weird chemical or agent in your environment. Now here's a pedigree, basically a family tree, right, of a family where childhood retinoblastoma is a problem. So if you're not familiar with these pedigrees, everybody marked with red had childhood retinoblastoma. And it's estimated about 40% of retinoblastoma cases are familial, and typically you can distinguish between familial and non-familial because in the familial cases you'll get tumors in both eyes, whereas if it's sporadic, usually you're just going to get retinoblastoma in a single eye. But this pedigree is interesting because if we look at this and consider, okay, does this trait look like it's being inherited in a dominant or recessive fashion? I would say at first glance it looks dominant, and the way you would distinguish these is usually if a particular trait skips a generation in a family, that would usually indicate a recessive inheritance pattern, whereas if it does not skip generation, then you'd say it was a dominant trait. And for the most part here, we're seeing it's not skipping generation. It's going directly like parent to child, right? Same thing over here, parent to child, parent to child. The one exception we see is right here, this guy, right? So, his dad had it, he didn't have it, but then two of his kids did. That would be more of a recessive pattern. But overall, looking at the family, it looks pretty dominant. So this would be an example of incomplete penetrance, right? Overall, this seems to be the dominant trait, but this one person is not really fitting the mold here. Would suggest that they don't have the phenotype that their genotype would suggest. So what's going on? Well, it's actually interesting because, yeah, this is inherited in a dominant fashion, but in actuality, it's really a recessive trait. It's due to recessive mutation, meaning both copies of the gene have to be inactivated in order for this cancer to develop. And so the gene that's involved with retinoblastoma is the retinoblastoma gene, right? Identified, first named because of this association. And the way this works is via what's called the two-hit model. And so essentially, in a nutshell, what happens is you would inherit a mutated copy of the retinoblastoma gene from either mom or dad, in this case from dad. So the little red spot here is supposed to indicate the mutated version of that gene. So now in this child, every cell in its body has one mutated copy of retinoblastoma and one normal copy. Right? And so in order for retinoblastoma to occur, for the cancer to occur, we need to have both copies inactivated. So what would have to happen is sometime over the course of their life, and usually this seems to happen pretty early in childhood, at least one cell in the retina has to pick up the second mutation, the second quote-unquote hit, so that now both copies are inactivated, and we get that cancer developing as a result. Now, this would be in contrast to sporadic retinoblastoma, where you don't inherit any mutations, right? So every cell initially is normal, two functional copies of retinoblastoma. At some point, maybe you pick up a mutation in one copy, and then it would require a second mutation in one of those already mutated cells in order for the cancer to develop. So you'd have to pick up, due to random chance, the two hits in the sporadic model versus you basically have a head start down that path in the familial situation. So again, it all comes down to needing those two mutations, which would make it a recessive syndrome. And if we do a little math here, I haven't done the math, somebody did this math. There's estimated to be about 10 million cells in the retina. Mutation rate is thought to be about one mutation in a million per gene for each cell division. And what that works out to in the real world is if you had inherited one mutated copy, there's now a 90% chance that the second copy is going to become mutated in at least one of those cells in the retina. And as we see, because this is usually a childhood cancer, it's in fact, a pretty high chance that that's going to happen pretty early in your life as well. So only about 10% of individuals don't pick up the second mutation. And again, just to reemphasize, this is an unusual example because in terms of the genetics, we would say this is a recessive cancer, right? You have to have two mutations to get the cancer. Both copies of the gene have to be inactivated, but it's inherited in this dominant fashion because the majority, right, 90+% of people are going to pick up that second mutation during their lifetime. Okay, so what is the retinoblastoma gene or the retinoblastoma protein do? Well, this is a classic example of what we'd call a tumor suppressor gene. Remember, tumor suppressors are proteins that inhibit cell proliferation. And retinoblastoma does that by acting at one of the cell cycle checkpoints, the restriction point before S phase of the cell cycle, basically preventing a cell from beginning DNA replication. And the retinoblastoma protein does that by binding to this transcription factor called E2F and preventing that transcription factor from entering the nucleus and activating transcription and hence translation of the genes and proteins needed for DNA replication. So in the normal situation, we would only move past that checkpoint if cyclin CDK shows up, phosphorylates the retinoblastoma protein, causing it to now release the transcription factor. But in our hereditary cancer, right, you have basically inherited one mutated version of retinoblastoma. So you have one copy of the protein now that's not working. You do still have one good copy that's enough to keep you healthy. But once that second copy is mutated and the protein inactivated, now you don't have any functional retinoblastoma, and this transcription factor is just free to always go into the nucleus, and you're just always going past that cell cycle checkpoint, even if cyclin CDK don't show up to phosphorylate. So you've sidestepped the red light, you've cut the brakes. Now, I also want to point out that the retinoblastoma gene and that mutation, right, you inherited that, or these individuals inherited that mutation in every cell in their body. So you could pick up a second retinoblastoma mutation in other tissues, right? It's not just in the retina, although that is where it was first discovered and where the name originated. But for example, we see about 20% of prostate cancers, 20% of breast cancers have inactivated retinoblastoma genes as well, right? So even though we're focusing on the retina, that's not the only place that it happens. And we'll talk about why is it predominantly in the retina. We'll come back to that in a minute. I apologize for the gross picture here, but the second example is a hereditary colon cancer, and this is the same kind of two-hit model we were just talking about. The gene involved in this case is this gene called APC, which is part of the Wnt signaling pathway. This is another example of a tumor suppressor gene, and we'll come back and look at what this signaling pathway does in a later chapter. But just know this gene would be tied in with slowing down cell cycle progression. And if you inherit one mutated copy of the APC gene, this is what your colon looks like on the left. It's basically covered with benign tumors, polyps, whereas this is what a healthy colon tissue should look like. And so if you inherited that one hit in the APC gene, it's almost guaranteed that by the time you reach age 40, at least one of those polyps is going to become cancerous. So really, the only medical solution at this point is just complete removal of the colon. So come back to this idea, yeah, we see in the case of retinoblastoma mutations, primarily it's linked to retinal cancer. In the case of these APC mutations, primarily it's linked to colon cancer, but again, not exclusively. We do see uh, increased risk of other cancers in those with APC mutations as well. So why is it that these tumor suppressor genes are primarily causing cancers in specific tissues? Not exclusively, but primarily. And really it all comes down to where are these tumor suppressor genes most highly expressed in the body. So you can imagine, if a tumor suppressor gene is not being expressed at all, right, not being transcribed and translated in some tissue in your body, then it doesn't really matter if it's mutated in that tissue, right? Because it's just not doing anything there. Um, so it's going to be in those tissues where that gene is and that protein is being produced the most that it's going to potentially be the biggest problem. And so what these cancers kind of reveal is, okay, well, the retinoblastoma gene, yeah, it's working as a cell cycle checkpoint regulator in, any cell in the body, but it seems to be most important for whatever reasons or most active in cells in the retina. And the APC gene seems to be most active in cells in colon tissue. So that's why we're seeing this particular link. But not always the case. So the third example I'm going to give you, called Lee-Fraumeni syndrome, is a result of mutations in another tumor suppressor gene, p53, which you may remember is linked to triggering apoptosis in response to DNA damage. And it turns out p53 is expressed pretty highly in lots of different tissues. So if you inherit a p53 mutation, there's a good chance that you could develop cancer in pretty much any tissue in the body. And in fact, this is particularly devastating syndrome because you'll see a wide variety of cancers, which makes it harder to predict what's going to happen and do any preventative diagnostics. But also you'll see individuals with multiple cancers during their lifetime. So this poor guy here had colon cancer at age 40, a sarcoma at 45, lung cancer at age 53. So this particular syndrome can be particularly devastating for a family. But again, right, just kind of speaking to this idea of where these tumor suppressor genes are expressed the most is where they're going to potentially have the biggest effect. So I just want to talk about some of the similarities we've seen with these first three examples. So they're all caused by a mutation in a single tumor suppressor gene, retinoblastoma, APC, p53. They're all following this two-hit model. You inherit one mutation, you have to acquire the second mutation during your lifetime, and the chances of that are pretty high typically, right? Because you had that head start with the first inherited mutation, there's a good chance you're going to pick up the second in some cell in your body. And a lot of times this may happen pretty early in life. So for any of these kinds of two-hit model inherited syndromes we've been talking about, each child is going to have basically a 50% chance of inheriting that same particular syndrome from an affected parent. So I'm going to stop there and we'll continue this in the second half.